Have you ever been tempted by those ads on TV to send a DNA sample be analyzed and find out more yourself? So that's Ancestry.com and 23andMe. So how many people have sent samples off to Ancestry? So a good number of people, okay. So perhaps whether it's the potential health issues in your family's origin, genome sequencing has definitely come out of the lab and into the marketplace. But there are a few things you might want to consider before sending a sample or spitting into a cup. We're going to discuss those today with a panel of experts, so I hope that you brought your questions. Genomics is a huge part, in fact, of biotechnology today, and we're pleased to invite the President and CEO of Ontario Genomics to help moderate our panel, Bettina Hamelin. So Bettina joined Ontario Genomics in August of last year, so welcome to Ontario, Toronto. Before that, she served as Vice President of NSERC's Research Partnership Directorate, where she was responsible for a range of programs designed to stimulate increased public-private sector collaboration and technology transfer by connecting the amazing Canadian research enterprise to Canadian global innovation stakeholders. Bettina has more than 15 years of experience in biotech and international pharmaceutical industry, as well as 10 years academic experience as a professor at the Faculty of Pharmacy at the University of Laval. Before joining NSERC, Bettina held a variety of leadership positions at Pfizer Canada, Canadian Medical Lead Vaccines, and head of strategic research partnerships in Western Canada. I've met with Bettina. She's passionate about R&D, and she excels at bringing together unlikely partners to attract and leverage funds from diverse sources. She's known for pioneering novel public-private partnerships models and breaking down barriers between federal, provincial, and private sector stakeholders. Early roots in biology and chemistry have led Bettina to complete a BSc in pharmacy and a Doctor of Pharmacy, both from the University of Kentucky, and an MBA from healthcare from the University of British Columbia, Canada. So please welcome me in joining Bettina Hamelin.
Thank you very much, Reinhard, for that nice introduction. Wow, it's wonderful to see all of you here this afternoon. I knew this morning hibernation is over. We need to get out. The sun was out, and despite the nice sun out, you're in here with us. But so I'm Bettina Hamelin. I'm the President and CEO of Ontario Genomics, and I'm just so thrilled to be here today and to be moderating a very intriguing, timely, and topical panel discussion this afternoon. Whose genome is it anyway? And this is a topic near and dear to my heart, not only professionally, but also personally, as you'll find out. You know, Reinhard read sort of through my CV. I've done a lot of different things, but a common thread throughout my career in both the private and the public sector has been dealing with new and emerging issues related to novel technologies and innovations. And the discussion today is really around that. So just to tell you a little bit about Ontario Genomics, in my current role there, I'm in the business of funding genomics-related applied research and the translation thereof in life sciences. And we're supporting numerous projects that are leading to some very exciting genomics tools that on the health side are working their way into the clinic, slowly but surely. But there's all kinds of organisms with living cells that carry genomes and DNA. And so we're also supporting projects in other areas to lead advances, for example, in agriculture, bioproducts, or even the mitigation of climate change. So today, I think we're going to get a glimpse into an area that just demonstrates how technology has really exploded in the last 20 or 30 years. And we've given ourselves some pretty exciting tools that allow us to sequence our entire genome. So looking around at the audience, I think there's many of you who are familiar with the terminology, but because we're going to be throwing some words around, I thought I just do a little bit of Genome 101, so just so we're all on the same page. You know, what is your genome? And you probably have heard of DNA. Maybe you've heard of DNA in school. Maybe you're watching Miami Vice, and generally the DNA evidence leads to the murder, except for with celebrities like O.J. Simpson, for example. But, you know, the DNA is a really elegant and complex molecule, and it carries all of the information that makes you function as a human being. And the genome is really just a description of all of that DNA in a living organism. It's the one thing that's similar or common to all of life. Whether you are a yeast, you're a mouse, you're a tree, you're a tulip, or you're a human being. We share that genome or that DNA. And the DNA has been around forever. It's been living in us for thousands of years, and that's where this whole idea of Ancestry.com comes about. There's a lot we can learn because it's been around forever. And I'm just going to leave you with a couple of kind of nice facts about genomes. For example, did you know that as a human, a mouse, and Pinot Noir, we all have about 30,000 genes. So it's good to know we are just as many genes as the Pinot Noir we're drinking. You know, take a look right or left or behind or in front of you, and you probably see that you all look very different. But let me tell you that all of us are about 99.9% genetically identical. So when we're talking about these genetic tests, we're looking at very, very minute differences between us. So the first human genome, of course, was published in 2003. It cost $3 billion. It took 10 years. It's now a lot cheaper and faster. And that's what's changing the landscape, and that's what leads us to having the discussions we're having today. So it's enabling personalized medicine, an emerging approach for disease prevention and treatment, and it considers a whole different set of information, genes, but also big data, environment, lifestyle, etc. And the hope, of course, is that personalized medicine can really improve our health care and help conserve some costs to the system. However, integration of personalized medicine in our healthcare system is no easy thing, and Wendy will be talking about that. But then the other whole thing is often referred to as leisure genomics, and that's where organizations like 23andMe and Ancestry.ca, those are probably the most well-known ones, but there's lots of others, offer tests for about $100 to let you know about your genome. So both of these areas, the healthcare-driven genomics and the leisure genomics, raise many perplexing ethical, economic, legal, and social issues, and that will be the subject of the discussion today. So that's the challenge, and let me get right into introducing our wonderful, esteemed panel. And I'm going to start with Dr. Fritz Roth. Dr. Roth is the Canada Research Excellence Chair in Integrative Biology at the University of Toronto. He completed his PhD in biophysics at Harvard University after studying both physics and molecular and cell biology at the University of California in Berkeley. After receiving his PhD, he gained experience in applied biotechnology as a scientist at Millennium Pharmaceuticals, and he currently co-directs the Genetic Networks program of the Canadian Institute for Advanced Research and is a member of the Center for Cancer Systems Biology at the Dana-Farber Cancer Institute in Boston. Michael. I think in the interest of time, I'll introduce all our speakers, and if everybody could come up here, and then we'll invite Fritz to give the first talk. Second panelist is Dr. Michael Szego. He is the director of the Center for Clinical Ethics, which is a joint venture between St. Joseph's Healthcare Center, St. Michael's Hospital, and Providence Healthcare. He's also an assistant professor in the Dalla Lana School of Public Health and the Department of Family and Community Medicine at the University of Toronto. Dr. Szego has a doctorate in molecular genetics and a master's degree in bioethics from the University of Toronto. And he lectures widely in both university and hospital settings and has research interest in the ethical issues associated with whole genome sequencing and the integration of ethics into clinical care. Dr. Szego. And last but not least, Dr. Wendy Ungar. Is a senior scientist in Child Health Evaluative Sciences at the Hospital for Sick Children's Research Institute. She's also a professor in the Institute of Health Policy, Management and Evaluation, and an adjunct scientist at the Institute for Clinical Evaluative Sciences. Dr. Ungar holds a master's degree in pharmacology and therapeutics from McGill University and a PhD in health policy, management, and evaluation from the University of Toronto. Dr. Ungar leads a program on research applying health economic methods to child health with a current focus on genomics and developmental disorders. She is the chair of the newly formed Ontario Genetics Advisory Committee with a mandate to make funding recommendations regarding emerging genetic testing technologies for the province of Ontario. Dr. Ungar wrote a book, The Economic Evaluation in Child Health, which was published in 2010 by Oxford University Press. Hello, Wendy. So welcome to Fritz, Michael, and Wendy here to the stage, and I'm really privileged to be with such an esteemed panel. By the way, I'm looking at myself here really of being just one from the audience. Somebody threw a coin and it turned out that I had to stand up here and ask the questions first, but you'll ask your questions. I think there's lots of questions about it. We asked previously, some of you already have analyzed your DNA. How many are thinking about it? Is anybody thinking about getting their genome tested? All right, there's a few of us, so you'll have to help us make our decisions. That's the question. So let's just go right into it. There's so many different options we have. We have these, leisure genomics. We have across the street a study that's going on, the personal genomics project. Where should we go to get our genome sequenced? And so let me start by asking you, Fritz, to tell us about that and what is it all about, and is it all whole genome sequencing? Is that what 23andMe does? They can do it for $100. That's amazing. So can you demystify that for us?
Okay, thank you.
All right.
Let's see if I can open the slides here to start things off. Okay, and let's see if this works. Hey, look at that. Okay, so I'll kick it off with leisure genomics. And so I'm a geneticist by day, but a dad nights and weekends. And so I thought it'd be fun. This is my rationalization of this, is to use it as a teaching tool for genetics for my kids. It's cheap enough that some of you would pay that for a day course for your kids in genetics. So I'm just, basically all I'm gonna do is in 5 minutes, maybe, I'll walk you through my experience with 23andMe, and you can decide whether that's worth your $100 or whatever it is today. And probably most of you will get a similar answer to what I got. But it's worth saying to start that 23andMe is not sequencing your genome. What they do is you spit in a tube, send it off, they get the DNA, and they only look at 700,000 positions in your genome, which commonly vary in the human population. So I say only because there are 3 billion that they could have looked at, and they only looked at 700,000. So that sounds like a lot, but actually they're only looking at the commonly varying positions. And in fact, there are 10 million positions in our genomes that commonly vary. But a convenient fact is that they travel in crowds together, so that if you only look at 700,000, you can actually figure out most of the 10 million, which of them you have. So that's what they do. But the caveat is that mostly, if a variant were bad for you, it wouldn't be common. So they're looking at the common variants, and the catch-22 is that mostly they're not informative. So maybe a couple of people in this room, if you did 23andMe, would find out that you went from having a 9% lifetime risk of late onset Alzheimer's to an 87% risk. That would be a bummer for a small number of people in this room, and there are a smaller number of people who might learn that they're carriers for a damaging breast cancer risk variant, but mostly you'll get answers that are similar to this. So I'll just go through some of the reports. So they give you, I don't know how to do that, but better? Okay, great, thanks. So you get an ancestry report, various other traits that don't have a whole lot of health significance, some wellness-related traits which have a little bit more significance, and then there's some health information that I'll show you that isn't in their standard list of reports here, but I was grandfathered in because I did it early. But anyway, so there's a smaller number of health traits that you can get information on. So I'll just step through it quickly and give you a sense. So here are the reports. And so my name is Fritz. My nickname is Fritz. My last name is Roth. You might have guessed I had some German ancestry. Sure enough, the light blue here is the predominating sort of Northwestern Europe component. No big surprise there. And French and German, okay, mostly German, but and they couldn't tell the difference, but I think they're doing okay so far, but no big surprises for me. This is where I'm way above average, though. I am more Neanderthal than 98% of all people who did 23andMe, so I'm pretty psyched about that. But actually that accounts for the Neanderthal ancestry accounts for less than 4% of my overall DNA, so I'm still not sure how that actually matters in a practical sense. Okay, my DNA relatives. So I could go find relatives, other people who signed up for 23andMe, and it turns out that my dad is really my dad. There's my dad. And that my 4 boys, are actually my boys. But actually, I knew that as soon as my then wife, said that it was okay to do 23andMe for everybody. So actually, I didn't even need to do the test. But I did, and they're my boys. And by the way, they all assented to my showing their names and a little bit about their genomes, although whether they have informed consent we can talk about later. They gave informed consent, yeah, maybe not. So go a little farther down and we see, hey, that's my dad's first cousin, Tom Wirt. That's cool. But I don't know. I mean, I knew he was my cousin, and actually, I don't talk to him much anyway, even if I didn't know that he was my cousin. And so the next guy is Mark Johansson. I don't know who that is, so who's that? We share 1.65% of our DNA. He lists some surnames in his family and where his family's from. And hey, my great-grandmother's maiden name was Botzloff, which is a pretty unusual name, and she was in Minnesota. So I think they got something right. I haven't figured out exactly how we're related, but I think it makes sense. And that's, we had a quick exchange. Yeah, that makes sense. And then, who goes to talk to their third cousins all the time? I mean, that's it, right? So it's kind of fun, but, and I can smell asparagus odor, and I don't have a lot of back hair, and, okay. So there's some other things that I knew that they got right, and that's kind of cool. And yeah, no cheek dimples, bummer. And okay, so they also guessed that I like to drink a little bit of coffee, and anyway, so they got some things right. Here's some bad news and some good news. I have an increased risk of atrial fibrillation and a slightly increased risk of some rare things that anyway, none of these are actionable. I don't think I'm going to change much about my lifestyle from these increased risks, but it's interesting. My biggest gripe with all of these health information statements is that they don't give me how sure they are about this risk. Because sometimes their uncertainty about that risk ranges from 27.3% to 40%. And I don't know how sure they are of that number even. And so that's one gripe I have about this. But basically, nothing life-changing here. I can also blame my genes for being a little more heavy than average. So this is sort of the fun. You can rationalize yourself this way. That I'm able to drink more, I'm likely to consume more coffee, and I'm likely to have a muscle composition that's uncommon in elite power athletes. So that's kind of letting me off the hook. That's cool. So interestingly, so this is if you dig in on this variant, so I have the TT version. So of course we each have 2 copies of each chromosome and each gene and each base. And I am T at both positions, both copies in this position. And so basically if I had a C variant, either 2 Cs or 1 C, then I would make this protein called alpha-actinin 3 that is found in fast twitch muscle fibers, and I might be more likely to be a sprinter or a weightlifter. But I don't, so that's okay. There's some, I think, sketchy evidence that I'm at greater likelihood of being an endurance athlete. I don't think actually the evidence is good, but I feel good about that, and kind of bad that I'm not an endurance athlete, but okay. So now let's look at my ex, who, by the way, agreed to be talked about today, and agreed to have all the genotyping done. And so this is me, uncommon result for elite power athletes. This is my ex, and she has 2 black belts in martial arts, and so maybe this makes sense for her that she has this common result in elite power athletes. And the genetics is actually predictable sometimes. So I have 2 Ts, she has 2 Cs. We each give 1 copy to our kids. So hey, look, all of my 4 boys have CT here, and so they're in luck, or at least on the hook, to be power elite athletes. So there you go. That's the power of genetics. And so some comments at the end of this. This was the quick, I hope, 5-minute tour of 23andMe. It's fun to think about, but for most people, you won't get information that's very predictive for you. So the common variant analysis has been incredibly useful to link specific genes to specific diseases when you take a large population in the aggregate. But it's not very useful to make something predictive, a statement about your health risks, usually. But it feels good to share your genome for research to find associations. The big focus of my group, so I'm a geneticist by day, and are these rare variants that have bigger effects, but we can't do statistics because we don't see them often enough. And so we actually have to test, does this variant do something? And so that's what my lab is focused on. If there's more time to say more, I will. And the other final comment from this is that sharing your genome, as you see, can reveal information about your relative's genome, and maybe we can ask some of the other people on the panel about what are the ethical implications of that. So I'll stop there and sit down.
Terrific. Thank you very much, Mike.
Okay. Oh, excellent. Get out of there. Okay, I will just close this. Okay, hi everybody. So I'm going to try and sort of map the issues, I guess. So to do a 5-minute lecture on bioethics or a 5-minute lecture on the ethics of genomics is impossible. So I thought I'd tell you some stories. First I'm going to tell you, I'm going to give you a little bit of a background. I'm going to talk a little bit about how your genome is identifiable, right? So when we talk about privacy and your right to privacy, that's really incompatible with genomics because your genome is unique to you. Although we're very similar to one another, there are differences, and those differences can identify you. So I'm going to tell you a little bit about that. I'm going to use the Personal Genome Project Canada as sort of an example of how we sort of operationalize some of these issues. And then I'm going to tell you a little bit about my genome because I was the second participant in the Personal Genome Project, and I actually had my genome done. So I'm going to give you sort of a tale of 2 variants. Let's see if I can do that in 5 minutes. So what is bioethics? Again, it involves critical reflection and about moral and ethical problems in healthcare, in research as well. And it's really about deciding what we should do, explaining why we should do it, and describing how we should do it. So I work with patients in hospitals often, a lot of end-of-life decision-making, and then I have sort of one foot in the research realm where I'm thinking about these issues in a whole genome sequencing context. So one kind of interesting thing whenever I go into sort of an ethical encounter is people say, well, I just want the answer. And so ethics isn't about me coming in and providing an answer. It's about providing the tools to think about the problem and coming up with a justifiable answer. There's no universally accepted ethical theory. If there was, we could just put in the question into a machine, turn a crank, and out would come the answer. It's about deliberation, and it's about coming up with reasons why we think a given course of action is the right way to go. So here's just a couple of principles. And again, I've sort of tried to tailor this to the genomic context. So sort of general principles in medical ethics, sort of on the left-hand side, we've sort of got the autonomy of the individual, right? So in a research context, we'd want to get somebody's permission before we got a blood sample and sequenced their genome and gave them back any information. We'd want them to know what we were doing with that information, what sort of privacy controls we would put on that information, what kind of data they would be receiving, who would be receiving the data, and those sorts of bits. We're also concerned about benefit. So in a clinical context, we want to make sure that the test we're proposing at least has a chance of benefiting the patient. Research is a little bit different. It's not about benefiting the individual, but it's hopefully about having some sort of scientific benefit. Otherwise, there's sort of no sense in taking on any risk. The other is about preventing harm. So in research, it's about trying to maximize benefits and minimizing harm. In the clinical realm, it's really about sort of making sure that we're not harming individuals. And I sort of have a privacy sphere because that's all, it is about preventing harm. So we want to make sure that identifiable information about you isn't sort of leaked out, causing harm. And we also want to make sure that individuals really are appraised about what the likely privacy implications of their decisions are. On the other hand, I've got public health ethics. So one of the interesting things about genomics is it kind of straddles the two, and I've got a case example about that, which is public health ethics is about balancing individual autonomy with the greater good, right, with the greater health needs of society. And because genomics is shared, right, so you share your genes closely with your nuclear family and then with your broader relatives, so genetic information about you has implications for your family. So it's not just about your rights to that information, but it's about, do you have any obligations to share that information to your family members? And that's when it gets a little bit more into the public health domain. So again, I'm just trying to sketch some of the issues here, and now I'll start telling some stories. So this was a case that I had when I was an ethics fellow. So I was at a major academic hospital, and I got a call from a geneticist. I was very excited because that was my background. And the geneticist said, I've got a problem. I've got a patient that tested positive for a colon cancer susceptibility gene. So this patient would have a very high likelihood of developing colon cancer over the course of their life. It's dominant, so you only need one copy of the gene, which means that this patient's siblings are also at a 50% risk of having that same mutation and therefore being at that increased risk of colon cancer. The problem was the patient didn't want to disclose that information to her family members. And the question that was posed to me was, can I trump the confidentiality of the patient to then inform family members that they're at risk of developing colon cancer? And that's when we start getting into, if we're using a conventional medical ethics paradigm, we'd probably say that autonomy trumps, so that the individual has a right to their own healthcare information, and therefore it shouldn't be shared. If we're starting to think about this more in public health terms, then the family would certainly have a right to have that information. So that's one story. The second is around privacy. So this was the 1,000 Genomes project was published several years ago. It involved sequencing 1,000 genomes. They eventually were able to come up with the ability to make it publicly available with consent of all the research subjects. So all that would be, if you wanted to look up any of these genomes, you would see the genomic sequence and the city of origin of the research subject. That's the only information you'd have available to you online. What a group did is they went and they took that publicly available information and then they cross-referenced it with ancestry databases. So I've picked on Ancestry.com. That's one of several companies that are doing this. And again, the whole raison d'etre for these types of companies is to have people identify relatives and see what their shared ancestry is. So by doing that, they were able to obtain a last name of certain individuals within the 1,000 Genomes project. Then they looked at the white pages, or Canada 411, or whatever the equivalent was, and they were able to re-identify about 50 individuals just using publicly available information. So there's nothing insidious about that, provided the research subjects knew that they could be identified. And I think what this highlights is that your genomic information in the right hands, with the right person with the right bioinformatics tools, you may be able to be re-identified. So again, it's not a showstopper, but it's something that people need to be aware of. The other question is, well, where does the data go? Right? So these are enormous amounts of information. When we're talking about direct-to-consumer genetic testing, often the information then goes to a database. And if you don't read the fine print, you might not know what that database will be used for. In the majority of cases, they're actually selling that information to third parties that may be wanting to do other experiments with it. And again, I don't know about you, but when like the Apple terms and references, you get like 4 pages of very tiny print. You scroll through it and you consent at the bottom. Well, contained to that information, when you sign up for one of these services, could be that we're going to sell your information to a third party. So again, it's just buyer beware. You need to know that it's not just about necessarily finding out what your ancestry is. It may be a tool for this company to be able to then use that information for other purposes. When we start talking about clinical sequencing, what happens to the clinical data? Does it just sit on a clinical chart? Could it be used for research? Ought they get consent up front for being able to use it for research? When we're talking about research data, should it be on a restricted access database where only researchers studying a specific disease can have access, or all researchers? Or should it be on an open database like the Personal Genome Project where anybody can have access to it? Which leads me to the Personal Genome Project Canada. So this was one of the research participants that first joined the project and agreed to be identified, and he was on the cover of the Globe and Mail, I think February 3rd. And this was Carolyn Abraham wrote a really great piece about sort of the sequencing and the analysis of the first 56 individuals. And that was published in the CMAJ as well. And so the intention of the project is for people to come in and make their data publicly available. So we consent individuals, we screen them, and we ask them, do you want to make your data publicly available? And if they say yes, then they can sort of proceed with the project. We'll sequence their genome. We'll make sure they sort of understand all the risks and benefits. There's actually an entrance exam. We put our research participants through a few more hoops than sort of the average research project. Then at the end of the day, we give them back a research report. Then they have to make the choice whether they want to make that data public or withdraw from the project. So we keep everything private until the research subject can then make that final choice. And we're clear, once the data goes out on the internet, we'll happily take it off of our servers if you want to withdraw from the project, but we can't mandate a third party to do so. So once it's out there, it's going to be out there for the world to see. So this is sort of a really brief schematic. These are all the different steps that one has to go through. And then at the very end, there's another consent to make the data publicly available. So we try and keep the research subject in the driver's seat to the best of our ability, really make sure that they understand and appreciate what the risks are, and then once they have all the information, to make that choice to make that data public, or again, to withdraw from the project. And if they withdrew from the project before making the data public, only they would know that they had their genome sequenced, because it would be off of our servers. And that's where I will almost end off. This is a copy of my genome sequence. So these were, unlike 23andMe, we actually look at the rare variants because we think that those are actually more important for decision making. So I was identified as having 15 rare variants. From a genomic perspective, I'm actually relatively healthy because we've been getting on average 20 to 25 rare variants per research participant. And I want to identify 2 that kind of grab my attention. So I still remember looking at my genome sequence. It was or my report. It was a year ago in February. Opening up my report, and I scanned down the list, and I saw hypogonadism, right? Which is, so hypo means less than average. Gonadism, you know what a gonad is. So that's not 2 kind of things you want to hear next to each other when you're talking about your. So I did a bit of, did a little bit of research and figured, so what is somebody who has hypogonadism? Like, what is that like? What are the traits like? They wouldn't have gone through puberty and they would have been sterile. So I've got 3 kids that look a little bit like me, so I don't need to get their cheeks squabbed like Fritz did. I'm pretty sure they're mine. And they wouldn't have gone through puberty. So puberty happened around the normal time, and I've got 3 kids. So I clearly don't have the trait. The key thing here is that it's almost kind of funny when I talk about it now, but if I had this done for one of my children prenatally, I'd be maybe very concerned about what their future life would be like, right? So you really need a phenotype. One of the things that we've learned in this project is that the data is only good as the databases, and the databases don't have complete information yet. So we get a lot of information like this that doesn't necessarily map onto the lived experience of our research participants. So until we get more genomes and we're better at predicting, we're going to continue to get sort of variants like that. The other is I'm a carrier for the Tay-Sachs disease. So Tay-Sachs, you get one copy of a gene from mom, 1 copy from a gene from dad. You need 2 mutant copies to have the trait. And it's a lethal condition in childhood. It's most highly prevalent in the Ashkenazi Jewish population. So I was raised in a Christian household, but my grandfather's Jewish and actually survived the Holocaust. So I suspect that I got that trait or that mutation from his lineage. And when I first saw that, because I'm a geneticist and I knew a little bit about that without having to do much digging, my first response wasn't that I was sort of upset that I had this trait. I actually felt like this was a manifestation of my family history. So I felt, not pride is a bit of a strange word to use for this, but that probably comes pretty close to how I felt when I saw that trait. Having said that, that's something that I would inform my kids about because they're going to be at a 50% risk of having that same mutation. And depending on who they partner with, if they decide to have kids, that's something they're going to want to know about. So this would be in a category of, for reproductive decision making, it would be important, but it also speaks a little bit to my ancestry and my family history. So now I will end there. So I think I went over my 5 minutes, so sorry about that. But thanks for your attention. I look forward to some questions. I think we've got Wendy next. I'm not sure which one you are, so I'll let you.
Okay, well, thank you very much. Before I begin my talk, I just wanted to make a few disclosures. Although I am chair of the Ontario Genetics Advisory Committee, which makes funding recommendations about genetic testing for the province of Ontario, all my views and opinions expressed today do not represent Health Quality Ontario or the Ministry of Health or Hospital for Sick Children or University of Toronto. They're just my views as an independent research scientist. The second disclosure I want to make is, unlike my esteemed colleagues, I have not had my genome sequenced, nor do I have any interest in doing so. So I represent a very broad spectrum of opinions on that subject. So what I want to talk to you about is in the medical arena, in our healthcare system. We're hearing a lot about the kind of recreational side of genomics, and also all the exciting research that's going on, which is fantastic. And of course, I'm highly supportive of understanding the variants and how they predict disease and health. But when it comes to the real world of providing healthcare services, we have to understand, how do we get these tests actually into our healthcare system? How can a doctor order them? How do they get funded? So that's what I want to just give you a very simple overview of that process today. And it's a similar process for technologies and medications, but I'm just focusing on genetic tests. So in the province of Ontario, we have a committee that looks at genetic tests, and only genetic tests, all right? They're only interested in that. And the first step is that topic selection. So specifically, which genetic test should we look at? Should we look at carrier screening? Should we look at non-invasive prenatal testing? Or liquid biopsy, or whole exome sequencing, or whole genome sequencing? There are many, many different types of genetic tests out there for different types of target populations and different applications. So first, the first step, of course, is topic selection. And there's a portal on the Ministry of Ontario or the Health Quality Ontario website where patients, clinicians, researchers, manufacturers, anybody can suggest a topic. So all of these get put forward, and we then have a prioritization process, and we think about what's the demand by clinicians, what's the need, is there an unmet need amongst patients for particular types of services or tests. So we prioritize. We also think about who will benefit from these tests, what's the state of the art. So there are many different factors that go into that prioritization. And eventually, topics move forward. And when I say move forward, I mean they move forward to what's called a health technology assessment. This is a full program of research, for the most part undertaken by a brilliant team of researchers and analysts who are based at Health Quality Ontario in the Ministry of Health. And what they will do is they will do a very in-depth review of the literature to look at accuracy, because we're assuming that all these tests are perfectly accurate. Well, they're not necessarily perfectly accurate. So we look at accuracy, for example, rates of false positives, rates of false negatives. We look at cost effectiveness. What's the incremental value? What's the value for money, the bang for the buck? We also look very carefully at any literature that we can find that talks about how patients value these tests. What are their preferences? What's their willingness to know? What are their concerns? How much do they want this technology? And there's quite a bit of literature around genetic testing, around the values, not just patients, but parents as well. And we also do what's called a budget impact analysis. This is the Ministry of Health. This is taxpayer money we're talking about, so we have to be very careful with it and understand, well, if we were to fund this test in our healthcare system, what's the total budget impact to the ministry? Is this something that we can afford? Do we have to think about, if we're going to pay for this, is something else going to have to come off the table, right? So we do that budget impact analysis as well. So this full dossier of all of this information then goes to my committee, the Ontario Genetics Advisory Committee. And who's on this committee? Well, besides health economists, we have several medical geneticists. We have clinicians, primary care clinicians. We have ethicists. We have patients. We have parents. We have policy experts. So it's quite a large and precisely selected committee to represent a range of experience and interests, and we deliberate and we think about, well, we look at all of the evidence and then finally we make a funding recommendation. It's not a final decision, but we make a recommendation about whether this test should be funded in the province of Ontario. And right now, just to let you know, it's a new committee, and the very first topic, we're almost completed with our first health technology assessment, and it's non-invasive prenatal testing. Right now, it's paid for any women who are at high risk, women over 35, or if there's a history of genetic malformations in the fetus. But right now, we're considering, well, do we want to actually make this a funded service for all women in Ontario, all pregnant women in the first or second trimester of pregnancy? Because the alternatives, things like amniocentesis, can actually be quite dangerous. So there's this lovely genetic test, much lower risk to the women, and we're right now deliberating over whether it should be funded. So stay tuned about that decision. So once we make a funding recommendation, it gets posted on the website, and we invite feedback from the public. Anybody can go on and comment about this decision, can question the report. The report is made publicly available. They can question the methods, question the findings, and then we collect all that information after the feedback period, and we think about the decision again. Do they have any valid points? Do we need to tweak the funding recommendation or change it entirely? So it's a somewhat iterative process. And then the final recommendation coming from our committee goes to Health Quality Ontario, goes to their board of directors, and then they make a recommendation which finally goes to the Minister of Health. So that's sort of the process in general, and it's the same process, not just for genetic tests, but for any kind of drug or device or technology in the province of Ontario. All of us pay for healthcare through our tax dollars, right? It's not an unlimited budget. And so the Ministry of Health wants to be in the position of funding those technologies that have the greatest value for money. So, we hear a lot these days hype about the $1,000 genome. How many people actually think that the cost of genome sequencing is lower than $1,000? Anybody? There's some people, yeah. How many people think that it's higher than $1,000? Well, there's actually about the same number. So I'm going to give you another number, $26,000 or $26,020. That's a value for money number. So what we're concerned with is not the price tag, it's not simply the cost, it's the value for money amount. And what does that $26,000 mean? Well, that's what it would cost to find one more child with a variant that predicts autism compared to what we're already spending today. So we already are doing tests for kids with autism to look for variants that explain where they are on the autism spectrum. We're already doing something called a chromosomal microanalysis or chromosomal microarray, which looks at the chromosome. So there's already genetic testing that's paid for in the province. So that $26,000 number, that's the additional cost above and beyond what we're spending today, it's paid for today, to find one more child with a variant that we'd want to pursue. So thinking about it that way, it's bang for the buck. It's given what we're already spending today, if we were now to fund genome sequencing, what additional benefit is it going to buy us above and beyond what we already have? That's the question that we're really interested in terms of decision making and policy making. It's not the price, it's not the price tag, it's the value for money. So if we're already doing a really good job with microarray or panels and cardiomyopathy or single gene tests, there's many different types of genetic tests that are perfectly fine for predicting risk of particular conditions. Maybe genome sequencing won't offer very much. So maybe it's better to spend that money on some other aspect of healthcare where we can get better value for money. So it's all about the value for money. I just wanted to make that point. The other kind of interesting and partly scary aspect to whole genome sequencing is there's a long list of potential variants that a patient might consent to have disclosed to them. So it's not just the target condition, it's not just the risk for a certain disease that they come into. It's a long list of other reportables. For example, BRCA1, higher risk of breast cancer, or higher risk of colon cancer, or Alzheimer's disease, or the Huntington's gene. I mean, do we really want all this information? Maybe some do and some don't. But what we have to think about, again, in terms of what our healthcare system can afford to provide, is that now, if you identify higher risks for these variants, what are you going to do? You're going to have your physician call up the specialist and make an appointment. So it's going to trigger all of these other healthcare services as you line up to get all of these other healthcare services. And in many cases, very important that you do get monitored and hopefully lower that risk, or even take some type of action that can prevent disease. So it's not just the test that's going to cost us, it's all of the other health services that are triggered as a result of the test. And not just the patient, other family members as well. So I'm in pediatrics. If a child is found to have a positive variant, the very next person that's tested are the parents. Where did this variant come from? Was it inherited, or is it what's called a de novo mutation that just arose in the child themselves? Need to know, because this might be something that's really important for the parents for future family planning. It may confer a higher risk in, let's say, a future sibling. So very important information to have. So there's lots of these sort of cascade triggering effects from whole genome sequencing that we need to think about. It's not just a single test, single result, single patient when it comes to this area. So I think I'll stop there, and maybe we can get into some other interesting questions. Thank you.
Excellent. Thank you very much to all three of our speakers. Obviously, they're all very passionate about the subjects they are covering, and I don't know about you, there's just so many questions. I'll maybe just kick it off, and I'd like to go back a little bit to the 23andMe story. And so if I followed you correctly, 23andMe does not do whole genome sequencing. It is a sort of SNP analysis. We're looking at certain variants that have been associated in large populations with certain disease traits. And you said, well, this is kind of fun, you know, not taking it too seriously. But you do get some numbers in your report. And I'm just curious if you could explain briefly, you know, what some of these numbers actually mean. So I saw in your report, for example, atrial fibrillation. I'm interested in that because I think my family has a predisposition to that. But when you go from an average risk of 27 to 33.9, what does that actually mean?
Right. So the point I made was that I'm not sure that 33.9% is the right estimate. That's their best guess, but I don't know how uncertain they are. And if you go back and look at the primary literature, I haven't done that for atrial fibrillation, but there are other traits. I should have mentioned, you can download your 23andMe data and upload it to other sites that are far less reluctant to give you information because they're not on the hook. They didn't genotype you. You came to them with a genotype, and they can say anything they want, and it's not a genetic test, and they do. And so I went to this other site with my data, and they told me far more shocking risk numbers than that for other traits. But I dug into the literature on those, and there was a case where your increased risk is anywhere from 1.1 to 300-fold. That was their range of uncertainty. But their best guess was 30-fold. And so all this second company told me about my risk was it was 30-fold higher. So anyway, I want to take these risk numbers with a grain of salt. But if that were actually my risk, I don't know what I'd do with that, right? So is it actionable? Would I change something about my lifestyle for something I'm likely to get? I think their cutoff is age 50. You know, are you going to get it before that age or not? Because it's a moving target, the older you are, the more likely you are to get some atrial fibrillation. And actually, atrial fibrillation is not dangerous in and of itself, except it leads to an increased risk of strokes. So I don't know. For me, that didn't tell me anything.
Okay.
That's it.
So can I take this report and take it to my physician and ask my physician what does that actually mean, and would I get that kind of answer?
My physician, I actually mentioned this to her, and she would just refuse.
Okay.
She just, unless it really looked like a smoking gun, but she just says she doesn't have the capacity or the training to go through this giant book that would be the results that someone could potentially bring her from a genome sequence. So.
Okay. Interesting aspect that I think both of you brought up, that these organizations actually share the data, and they sell it. I think, Michael, you spoke to that, and they sell it for research purposes, which is probably a good thing because we need to enrich the data. But how concerned do I have to be about them selling the data and perhaps telling me, but probably not telling me?
And I think it's about reading the fine print. So there was a study that was done looking at, I think they looked at 30 different direct-to-consumer genetic testing companies. I think only 10% of which would sort of promise that they would just keep the data for the purposes that you signed up for, which is to get ancestry information or to get some other piece of information. So 90% of companies would then potentially use that information for other purposes. Now, if you were to sign up for a sort of conventional research project, if genetic testing was sort of specific to the research, then that would be covered under the main consent. If they wanted to do genetic testing sort of as a side project, you'd usually have to sign another consent. And that's something that most companies don't provide. So usually you'd have to read the fine print to determine what they're going to do with your data versus being really, really transparent about what they're going to do with the data. So I'm not sure there's been any circumstances where a company doesn't disclose it. It's just really incumbent on the participant to make sure they're reading those 5 pages of legalese in 8-point font really, really carefully. So I'd be concerned.
You'd be concerned, okay. That's good to know. And is there any legal protection around the data in Canada?
So there's new legislation that protects people from genetic discrimination. So the U.S. had legislation actually going back to President Bush's days, GINA, and Canada recently enacted legislation which is very similar, except it actually is more inclusive. So as it stands right now, an insurance company could not use your genetic information to deny you insurability, to deny your insurance. What they will do is ask for family history. So normally, if you have a family history of breast cancer, which may or may not be confirmed using a genetic test, that's what they're going to actually go on. So I'm not actually sure that it prevents employment insurance, and that's probably the most important part. But in terms of insurability, I still think it's about family history, and you certainly would still be denied insurance because of a family history.
Okay, so there is legislation, but it's not that helpful because there's other discrimination.
Right, and again, in this context, so if a third party was using the data, I'm not sure that would ever cause insurance problems for an individual. It's more of what are they doing with the data as an autonomous individual. Do you want your data to be used by big pharma to be asking questions without your consent? That would be more of what I'd be concerned with.
Yes, okay. I'd invite you if you have any questions, there's 2 microphones, and I see. There's a microphone just behind you.
I got a question that I don't really know how to put into words. Could I show you?
Barry, can we do that after, please? Here.
It's just this one. Okay. It's just that it says that identical twins have a 52% chance of developing cancer. And I was trying to talk to Mukherjee, who wrote the Emperor on Cancer and the Gene, and his mother's is identical twin, and her sister suffers from, I guess, a split brain, and it's very difficult to put into words. But like, have they actually found out why this happens? Like, I was talking to James Till, and he says that it happens in the animal world all the time. It's only in humans that are able to talk and express, how they feel in that.
So I think this may be a specific enough question that we should either take it in the discussion after the session, or maybe it may be if it's a very specific to your family question. I'm not a trained genetic counselor, and I think it actually wouldn't be ethical for me to make a diagnosis here on the stage or to give you advice without hearing all of the details, and I'm not qualified to do that. So maybe we can we push it to a reception period afterwards? Thank you.
Other question? Oh. Do you have a question over here?
Oh, she's running the mic.
How do you join the personal genomic project, and is there any cost involved?
So there's no cost, and we need to register on personalgenomes.ca.
Other questions? Oh. Sorry.
No.
Just here and then you right after. Sorry.
So when you sign up for these programs, you're consenting for your information to be available to your companies, but what about the information of your family members? Do they also have to get your consent afterwards? Is their information is also available to them, or is that included in whatever terms you sign on to?
So the question was around when you consent for a research project, a genetic research project, are you also consenting for your family's information? Are you talking about the personal genome project specifically, or just in general? So in general, it's about the individual making an autonomous choice for themselves about whether they want to participate or not. One of the unique features about the personal genome project is, of course, the data is going to be made public, so that has implications for family members. So we do ask research participants to talk to their family, but they don't need to get consent from their family members, because you as an individual can make a choice about what kind of research you want to be involved in.
But 23andMe does not require consent from your family members to share your data too. So that's one big example of they say quite clearly, you can volunteer to participate in studies, and you consent to participate in studies that they will publish, but for things that they sell and they promise will not be published, they'll simply sell your data in the aggregate. They don't. Basically, everybody's data gets to be used, but it's de-identified, right, presumably.
Yeah, yeah. I think there was a question up here, the second row.
Yes, hello.
Hello.
Yep.
Yes, given that it looks so non-predictive, like there's so many variances, and the probability is pretty well unknown across the board, it looks like a more speculative science than anything else right now, genetics. So do you see a day where it actually improves a lot, the databases get a lot better, and it actually can become predictive as opposed to it's a bit hairy-fairy right now?
Right. I mean, I should say that sometimes it is very predictive. It's just a very small fraction of people who will get an answer that is both highly predictive and actionable. But there are people now, today, even with 23andMe, even just the common variants, I tried to make the point that some people will get an answer which is predictive for them, but mostly not. But for the rare variants, there was, let's see, I think there are about 4.5 million variants that we've seen that change something about a protein and so are potentially of concern. 2% of those have been looked at by a clinical geneticist, and only of the ones that have been looked at, more than half, the answer is variant of uncertain significance. So for the rare variants, we mostly can't say much with confidence, but the thing is we know that's where the action is. It's the rare ones that haven't, if the ones that are really damaging haven't had time to sort of, so if it wouldn't become common if it was really damaging to you, because there would be an evolutionary disadvantage, but you can find rare variants that do harm, and those are hard to interpret now, and that's, I do see a day when we can be better at that, and I'm working hard in my group, and for 8 genes so far, we've done experimental tests in a model system, but we've tested the majority of all possible missense variants and done an experimental test. Does that mess up the protein or not? Because just knowing there's a variant there doesn't tell you whether it damages the protein. And so for 8 human genes, we have a lookup table where we've done the experiment in a model system, a yeast model of the human process. And so but I think only in 3 cases has one of those been used in the clinic, has our table been used in the clinic. It's very early days.
If I could also just add to that, I mean, we're seeing this in pediatrics a lot, They're children on what are called these diagnostic odysseys. They're kids with complex conditions or very, very rare disorders. You know, there may be only a handful of these rare variants in all of North America. And so the parents have been seeking a diagnosis for their child for sometimes decades, and they have no answer. And for the first time with whole genome sequencing, they have an answer. They found the variant, the causal variant. Does it lead to different treatment? No. Does it mean anything different in the management of that child's condition? No, but the parents actually get an answer, and just having that information, whereas before they didn't, provides a lot of reassurance. And the other thing, it can actually save money. So if we know which children, or not just children, but adults, individuals with rare diseases, if they're morphologically complex, they have unusual symptoms that can't be diagnosed easily. If we sequence them, instead of putting them on this odyssey of test after test after test, in those situations, it's actually cost-saving to do genome sequencing. The other aspect, there's a parent on our advisory committee who's been in this odyssey for a decade with his daughter. She's 16, and finally, after many, many, many years, after sequencing at sick kids, they have the causal variant. And not only does it provide reassurance to the parents, now the parents can connect with the 5 other families in North America who are struggling with this and have sharing information, sharing of experiences. So there's value there that is hard to quantify because it's not necessarily leading to treatment, but there is importance in the information itself.
Okay.
Yes.
My question is,
Yes, question in the
Ancestry
back.
or 23andMe, most of the time the people are going to tell that you are 20% Italian or African or something like that. Is there any use or? Okay. Is there any use with that kind of getting information about your personality as an
Personality testing.
Yeah, so actually, I don't think I got any of that information, but I know they ask personality questions, and so they are certainly using the data to try to identify links to personality, and because they have a whole question series that they're basically, I think it's a wonderful feeling to contribute to these studies, and then you can go look on the site and see what conditions they've linked which regions of the genome to. And so they're looking at that, but I didn't get any results, and I just don't remember if that's something they're looking at now in reading your genotype if you submit it today. But it is a fast-moving target, and they actually are learning new stuff by aggregating questionnaires from all of the patients, all the customers, I should say.
Okay.
Yeah.
My second question is, okay, you are studying about a person, about his genome or any kind of disease he's going to get in future, right? But you are not sure about how much he is going to get the disease, right? The accuracy of the disease, we are just it's calculating by DNA sequence, right? But you are not scaring the patient by giving him information about some kind of disease or inability or thing.
I guess I couldn't make
Well,
that. Can you ask in a different way, maybe, sir, or
Okay.
or
When
just
we are studying the sequence of a person, we are studying about everything like his abilities or capabilities, everything, right? But there is a choice of he's going to get paralyzed or kind of paralysis or some kind of disease he may get in future, right? You are studying about that, right?
Yeah, I mean, they're trying to predict your genetic risk for,
Yes.
for traits, and some of them are bad. Yeah, sure.
But by giving that kind of information in future, I'm going to get after 20 years, I'm going to get some kind of disease. Whether you are not sure about it, you are giving some kind of scaring information to the patient.
Yeah. Yes.
Yeah, it is. It can be
Yeah, it
scary,
can be
and I think
scary.
this is a major. I mean, this is really a question for Michael. Yeah.
Well, I mean, again, I would say that's why genetic counseling would be important. That's why the informed consent process would be important. So even on this panel, Wendy said there is no way I want my genome sequenced, right? And I respect that opinion. I have a different opinion because I got my genome sequenced. So I had to think about if I found something in my genome that may be a surprise, would that be harmful to me? And I had to make a judgment about whether I felt that was okay or not. And I decided that I would probably want to know that information, whether it was bad or good, and I moved on from there. The other thing is the literature around people that have been disclosed damaging information. So for example, whether they have the Huntington's variant, which will lead to a neurological disease, shows that it actually, disclosing that information to them isn't harmful. they all had genetic counseling and they knew what the likely outcomes of the test would be, either they're going to have the variant or not. But the long-term outcomes weren't bad for those individuals. So I still think we need more information, but it's, I think, about making sure that people are really well informed and being attentive to the fact that it's still early days and we should still be studying it.
So related to that, can I just ask, are there things that you could opt out of if you give, for example, you're sending in your sample to 23andMe or whoever it might be? Can you just say, well, I don't want to know about these scary things like Alzheimer's disease?
So in the Personal Genome Project, we actually wouldn't accept somebody like that because you're making the data public. You can't say, I want to have certain pieces of information not told to me. So it's either you're all in or you're not in.
Okay.
In the majority of, but that's a very exceptional circumstance. In most genome projects, you absolutely have the right to get the information or not to get the information. Clinically as well, you may have colon cancer in your family, and you may be at risk for colon cancer, and they might want to do a genetic test on you, and it's about having a conversation to see whether you want to know that information. So you also have a right not to know.
Not to know.
And 23andMe does say that we're not going to tell you whether you're likely to get late onset Alzheimer's based on the ApoE4 variant unless you say specifically that's information you want about yourself. So,
Okay,
yeah.
okay, great. So one more question.
Hello. Wendy said that if they do this testing and they find these variants, they get information, but I think you said that the treatment doesn't change. And so I'm not clear on where the cost savings are coming from. And secondly, if there are these cost savings, are doctors commonly asking for these tests to be done, or is it solely the discretion of the parents and they have to pay out of their own pockets?
Good question. So I would say for individuals who have been on these so-called diagnostic odysseys, where there's just no diagnosis, the doctor can't find the cause of their symptoms, and they're getting test after test after test, then it would make sense if they had sequencing at the beginning and they could find the causal variant, it could save money because they would have avoided all those other tests. The difficulty is in identifying those patients in the first place to know that they would have eventually been on this odyssey. So for patients with rare diseases or patients who are certainly children with complex presenting symptoms where there's no obvious diagnosis, then I think it makes sense to do sequencing. So where are we today with that? Well, just to be clear, the sequencing we've been talking about is research or recreational. There is no funded whole genome sequencing anywhere in Canada right now for clinical care, for patient care. Whole exome sequencing is a little bit different. So that's sort of a mini version of whole genome sequencing where they only sequence a portion of the genome, the protein coding portion. So they only do a fraction of the whole genome. Now in that situation, at least in Ontario, there are clinicians who are ordering whole exomes for their patients, and what they've been doing so far is sending the sample down to the United States to be analyzed. They have a very quick turnaround. They get the results within a reasonable time. And then either the patient or the clinician submits the bill for that sequencing to the province, and the province has been reimbursing those costs in the out-of-country pocket program or out-of-country program. But that's just a Band-Aid, that's a short-term remedy. Now the province, that's why we struck this committee, is because there's an increasing demand for exome and genome sequencing in clinical practice, so we need to make a decision about whether to fund it or not. So those are the decisions in front of the committee today.
Can I just ask one question? What the heck is a Neanderthal trait? Do you have do you swing from trees in the morning?
Frontal ridge.
Well, I mean, it's pretty obvious, right? No, actually, I don't know if any of those Neanderthal. So basically, they've inferred that there was interbreeding between modern humans and Neanderthals, and there's not much left of that sort of a remnant of that event in the human population, and it's not clear. Michael may know, I don't know, if any of those have been associated with actual human traits. So you can see this, you can say it came from Neanderthal based on the fact that we've sequenced the Neanderthal genomes, or one or two of them. But what does it mean for me personally? I don't think anything. Yeah.
Wonderful. I think in interest of time, we have to wrap up, and I think this is a nice final note, and I just want to say that I was very happy to see that Fritz and I had something in common, which is German ancestry. But I'm not so sure about the Neanderthal ancestry. Maybe I don't want to know about that one. But I want to thank the panel for wonderful discussions that took us from the leisure genomics all the way to what if whole genome sequencing is ready for prime time and needs to be integrated in clinical care, and how would that work? I think that would be it was a wonderful exposure of that. I learned a lot, I'm sure you did too. Thank you for your questions, and I think you gave us lots to think about. And on that note, thank you very much.
Great, great turnout. Thank you, everybody, for coming. Great questions. I think the panel is going to be able to stick around for a few minutes. I would just say no personal questions. These are not genome therapists. They're not going to help you with your issues, but if you've got more generic questions, obviously they're very interested. Very interesting panel, I think, and I really want to thank you for moderating it and for the panelists participating. And I think we've got something for you that's on its way out. Here we go. Great. Thank you very much. So again, round of applause for our panelists. Thank you. And we'll just a reminder, we hope to see some of you. How many are coming Tuesday night to the Fleming Award? Few. There's still some seats available. I encourage you to think about it, and then we'll see you at the next Sunday lecture to find out about dirt in your diet. So thank you very much.